Everyone to our bioelectrodynamics webinar. I would like to announce that this regular webinar series is supported by International Union of Radio Science, URSI, as well as by Engineering in Medicine and Biology chapter of the Czechoslovakia section of IEEE. And it's my pleasure to welcome today our speaker, Domenico Scaramozzino, who is from Italy. He received his PhD in civil and environmental engineering in 2021 at Politecnico di Torino, and now he's still now as a postdoctoral fellow. And he was also visiting scholar at Iowa State University in computational biology in the lab of Robert Jernigan. And his research interests are interestingly lying at the interface of structural mechanics, ranging from the buildings down to the molecules, so to the computational biology as well. I would like to also highlight that he's authored a very nice short book on waves in biomechanics, terahertz vibrations, and modal analysis in proteins and macromolecular structures. For those who are interested in the topics of today's webinar, I guess they will be also interested in book from our today's speaker. So that's for the introduction, Domenico. So now I guess the virtual space is yours. So please go ahead.
Thank you, Michael. Thank you for your kind introduction and again for your nice invitation. It was really an honor for me. And thank you everybody attending, and good afternoon or good morning or good evening, depending on where you're attending this webinar from. So as said, I'm Domenico Scaramozzino, currently a postdoctoral research fellow at Politecnico di Torino in Italy in the Department of Structural Engineering. And I was also a visiting scholar at Iowa State University. So today I will give a short presentation about one of my last research work about how we can be able to understand the protein functional dynamics and functional motions by application of some dynamic perturbations, specifically in the sub-THz range. So just to give you an idea of where I'm talking from, I'm currently in Politecnico di Torino in Italy, so in the northwest part of Italy. And from the other side, I was 2 years ago. Yeah, 2 years ago, I was visiting at Iowa State University in the lab, in the computational biology lab of Robert Jernigan. So this is a brief outline of today's presentation. So at first, I want to give you some kind of comprehensive introduction about the dynamics of proteins and also some perturbation methods in order to then talk more in details about the main topic of this webinar. So about the applications of random harmonic sub-THz perturbations to the protein structure. So at first, we will just briefly discuss about the protein dynamics as the fundamental link and bridge between the structure of the protein, so the 3D structure, and the biological function. Then I will show you some experimental methods, in particular Raman spectroscopy and terahertz time domain spectroscopy, which tell us that the main vibrational modes of proteins are expected to lie in the sub-THz range. And then we will finally switch to the computational side of the presentation. So I'm mostly a computational guy. So I will show you how you can compute and describe normal modes of vibrations of proteins by means of normal mode analysis, and in particular with some simplified coarse grain elastic network models. And then I will also show how you can apply some perturbations or some forces onto the protein structure to get some understanding about the protein conformational changes and the overall flexibility of the protein. And then, of course, we will dive more into the main topic of today's webinar. So, as you might know, protein biological function strictly depends on the sequence of the amino acids that you have, and it was already recognized in the, let's say, in the 20th century that the three-dimensional structure of the protein is key. So, is key for driving the biological function. So, a certain biological function can be dictated by a certain three-dimensional structure. But at first there was this kind of static view of proteins, in the sense that once you have the information about the structure, you straightforwardly have the information about the biological function. Well, nowadays the paradigm is more is a bit more complex in the sense that now the paradigm also includes protein dynamics as the fundamental bridging point between structure and function. So if we are able to understand experimentally or numerically how the dynamics of a protein is supposed to be, we might be able to better understand the biological functionality of a certain protein structure. Okay, so the structure is not the only information that we need to understand the biological function, but we also need to understand how this structure evolves over time. So basically the dynamics. There are a bunch of experimental methods to understand protein dynamics experimentally. Some examples, for example, are infrared spectroscopy. There are some good webinars actually in this webinar series in which some authors describe some of these experimental techniques. For example, if I remember correctly, there was last month a webinar from Dr. Kevin Robben from University of Iowa, giving a very nice presentation about two-dimensional infrared spectroscopy to understand the underdamping nature of low-frequency modes in proteins. Then we can also use some Raman spectroscopy to understand the vibrational modes of proteins, where you basically hit a sample of the protein with a laser and you try to get some information about the dynamics by looking at the shift of the scattered light. Again, about this, you can have a look at another webinar in this series by Professor Patrick Sennett, who worked a bit about Raman spectra in proteins. Then other techniques can be, for example, nuclear magnetic resonance, when you take a look at the variations in the magnetic and the magnetic behavior of nuclei within a protein, or also another very important recent technique is THz time domain spectroscopy. Again, about this, you can take a look at another webinar by Professor Andrea Markels from University of Buffalo. And this technique is, in my opinion, is very good because it enables you to have some clear understanding of the response of protein dynamics in the range of terahertz and sub-terahertz vibrations. And also we have some, nowadays we have some very good microscopy techniques. So far, microscopy techniques are able to give you some understanding about the dynamics of larger proteins. But for example, you can take a look at the conformational changes of some big proteins with high-speed atomic force microscopy, or also by using the single molecule FRET technology, which is basically technology where you apply some probes into certain position of the protein structure, and then by evaluating the distance between these probes, which emit light, you can have an understanding of how the protein moves dynamically. And also, just the last one that I want to show you is cryo-EM. So cryo-EM, in my opinion, is going to be one of the main techniques to take a look at protein dynamics probably in the next few years or decades, because it enables you to see, in a way, what happens in the biological environment. So you can have an understanding of the dynamical motions of proteins within the, for example, the cell environment. So now let's take a closer look at two specific techniques, spectroscopy techniques, to understand protein vibrations, for example, Raman and THz time domain spectroscopy. So here you have the principles of these two spectroscopic techniques. Here you have the principle for Raman spectroscopy. What you do is you basically hit a protein sample or a sample of a certain material with an incident light, with a certain frequency, and then by the Raman effect you have some scattering of the lights, and this scattering is having different components in frequency. So you have a very strong component which is called elastic Rayleigh scattering, which happens at the same frequency of the laser. But then the most important thing is that you also observe some shifted bands, the Stokes and anti-Stokes bands, which are informative of the vibrational frequency of the molecule. Okay, so the shift is directly related to the frequency of vibration of your sample. So in our case, for example, the protein. And this is the principle of terahertz time domain spectroscopy, where you basically want to apply some short terahertz pulse to the protein, and then you record the transmitted or the absorbed signal. And then, for example, by carrying out a Fourier transform, you can have an understanding of the values of the vibrational frequencies which interact with the main vibrational modes of the molecule. So why I want to show you Raman spectroscopy and terahertz time domain spectroscopy is because these techniques show us that proteins have some strong vibrational modes at low frequency in the sub-terahertz range. So this was, in my opinion, one of the first papers in 72 showing a very low frequency peak in a protein sample of alpha chymotrypsin. These authors discovered a peak at around 30 centimeters minus 1, so around 0.87 terahertz. And the most interesting thing about this discovery here was that you are able to find this low frequency peak in the protein, which is associated to a certain unknown vibrational mode, for a variety of different preparation of the protein sample, except when you prepare your sample with an SDS solution. So basically when you induce the chemical unfolding of the protein. So when you have your protein unfolded, you don't see this peak anymore. So this suggested that this peak, this low frequency vibration, should involve a global vibration of the entire 3D protein structure. Okay, so global vibrations of the proteins in this sub-THz range. And also in the following years, there were other experiments with Raman spectroscopy showing again some sub-THz low frequency peaks corresponding usually to global vibrations of the protein. Otherwise, when you find some high frequency vibrations, these are mostly related to local vibrations of chemical groups or vibrations of parts of the proteins like amino acids or side chains. Another technique that can give us some idea of the frequency range in which the main normal modes of proteins happen to be is THz time domain spectroscopy. This is one of the first works from Professor Markels, who showed who basically reported the THz spectra for different proteins, lysozyme, myoglobin, and bacteriorhodopsin. And as you can see by looking at this THz spectra, so the plots of the absorbance in the THz spectrum, you see that the main vibrational modes of proteins always lie in the sub-THz frequency range. Just to give you an idea, in centimeters minus 1 at terahertz is around 33 centimeters minus 1. So here we are dealing with the sub-THz regime. Okay, this work is also interesting. If you are interested, of course, you can take a closer look at the paper. In showing you what happens at the THz vibrational modes of proteins when you, for example, change the temperature. So when you decrease the temperature of your sample, there is a decrease in the THz absorbance of the sample, or also what happens if you induce a conformational change in your protein. There is some variation of the THz spectra, so in the vibrational modes, but this is something that you can take a look yourself into the paper. And then other interesting applications of THz time domain spectroscopy is to see what happens at the THz vibrational modes of proteins when the protein is not unbound, but for example is bound to a certain ligand. Here you see the effect of both temperature and ligand binding, or also when you have some mutation inside the three-dimensional structure of the protein. So the mutation is expected to change a bit the structure of the protein, and a change in the structure should reflect into a change of the dynamics. So you should be able to observe some change in the THz spectra as well. But this again is just additional information. The main point I want to make here is that the main vibrational modes of proteins are expected to lie in the sub-THz range. Okay, now let's switch to the computational side of the presentation. The main approach that you can use to understand the vibrational modes of proteins is normal mode analysis. Normal mode analysis is actually a general technique, not only used in the field of proteins, but we use it, for example, for buildings or bridges, because as you might have heard before, I work in a structural engineering department, so I worked with normal mode analysis also for evaluating the building vibrations. So the goal of normal mode analysis is to extract the vibrational and dynamical features of any structure in terms of the so-called normal modes of vibrations and the corresponding vibrational frequencies. And to obtain these two informations about the dynamics of any system, you only need the information about the geometry of your structure, the mass of your structure, and the internal potential energy, which depends on how actually you describe the structure in terms of degrees of freedom. One typical representation of the potential energy of a protein structure is this one, which is typically included in, for example, molecular dynamics simulation, where you have the energy content depending on the bond elongations, the bond angular variations, or the dihedral angle variations, or also some non-covalent interactions, for example, electrostatic Coulomb energy, and also the van der Waal interactions, which are normally described by using the Lennard-Jones potential. So if you are able to write the potential energy of your structure, you can build the so-called Hessian or stiffness in structural engineering matrix. So this is the matrix containing the second derivatives of the potential energy with respect to the variation of the degrees of freedom. So in this case, of the coordinates of the atoms of your protein. So in this case, you can simply write the potential energy of your system as a quadratic form of the degrees of freedom of your structure, so of the displacement, by means of the Hessian matrix. At this point, what normal mode analysis does is very simple. It's simply equating the internal elastic forces, which is simply the product between the Hessian matrix and the displacement vector, with the inertia forces, which, according to the second Newton's law of motion, is the product of the mass matrix and the second time derivative vector of the degrees of freedom, so basically the acceleration. So once you are able to write this equation, you can look for an harmonic solution of the problem, and normal mode analysis directly provides you the normal modes of vibration, so the direction of motions of your system, so of your protein, and the corresponding vibrational frequencies of each normal mode. And this is carried out by means of a simple eigenvalue eigenvector decomposition of the Hessian and mass matrices. So this is one of the first applications of normal mode analysis in the field of protein carried out by Go and collaborators. Here you see the computed frequency spectrum of a small protein, BPTI. And again, you see that even computationally, the main vibrational modes, so the force modes, lie in the sub-THz range. So here you see the main clustering of modes is around 50 centimeters minus 1, which corresponds roughly to 1.5 terahertz. So you see that the main vibrational modes of the proteins happen in this low frequency range and in this sub-THz frequency range. And you see here one vectorial representation of one of the low frequency normal modes of this protein. So this is the second low frequency normal modes of BPTI, found to be computationally at 0.2 terahertz. In the following year, of course, you had some additional applications of normal mode analysis. This is the work carried out by the group of Michael Levitt. Again, the frequency spectra of different proteins. Again, the main modes are in the low frequency sub-terahertz range. And again, you see here a vectorial representation of the normal modes with the corresponding frequencies. We are always in the, let's say, tens, hundreds of gigahertz. So we are in the sub-terahertz range. So we can go a little bit more with the simplification of our problems about protein dynamics, and we can employ the so-called elastic network models. So elastic network models were introduced at the end of the 20th century, at the beginning of the 21st century, by Monique Thirion and the group of Atilgan and Jernigan and Bahar. And basically, elastic network models are a representation of the protein structure as a simple network of elastic springs. So you see the representation of your internal elastic potential now is very simple because it only corresponds to the strength, so to the stiffness of each spring connecting atom I and atom J, and the corresponding elongation or compression of that spring. So it only depends on the variation in the interatomic distances. So this is the distance between two atoms in the deformed configuration, and this is the distance between the two atoms in the undeformed configurations. So in this very simplified approach, you can simply evaluate the Hessian matrix of your structure, which again you can see only depends on the springs strength and on the three-dimensional arrangement of your protein structure, and then again eigenvalue, eigenvector decomposition, and you find the vibrational frequencies of your system and the normal modes. The most interesting part about elastic network models is that the authors, researchers already realized that the low frequency normal modes from these very simple models, so from elastic network models, are often biologically relevant. It means that they are often found to point in the right directions of the experimentally observed conformational changes. So you see here one example in which you have a protein, LBI binding protein, which has an open form and a closed conformation. So here you see the representation of the vector displacements inducing the protein from the open form to the closed form. So this is the observed open to closed conformational change. And here is the results of the computation. So this is the second, in this case, low frequency normal mode from the elastic network models. And you see that they point in the same direction. So this means that the low frequency modes from elastic network models are informative. So they have something strictly connected to the biological function of the protein in dynamical terms. Here you have another example where you have LAO binding protein in a representation in terms of absolute displacements. So you see here the displacements in the second low frequency mode, which is the thick line, compared to the displacement of the open to closed observed conformational change. You see that there is a correlation. So the correlation is not 100%, is not perfect, but there is a very good correlation. So meaning that one single low frequency mode has a lot of information about the direction of the conformational change. You can also quantify this agreement between each normal mode and the observed conformational change by means of this overlap here. So the overlap is a simple, basically normalized dot product between the vector of each normal mode and the vector of the conformational change. Here you see the results of this overlap calculation carried out by Tama and Sanjuan in 2001. And here you see basically how they were able to find some very good correlation. You see here 0.83, 0.84, 0.86, some very good correlations between one of the low frequency normal modes and the detected conformational change. I want you to notice two things from this slide because this will be important for the later part of the presentation. One is in this table. So it's always easier to get good numbers here, so good correlations between one of the low frequency mode and the conformational change when you calculate the modes in the open conformation. When you calculate the normal modes of the closed conformation and you try to simulate the reverse pathway, so the closed to open conformational transition, the overlaps are consistently lower. And this is mainly related to the intrinsic ability of the elastic network models to capture better the dynamics of some more open conformations than the closed ones. And another important point about these results here is that if you look at this table, the good overlap values that you are able to find are more or less always correlated with the collectivity of the conformational change. So this means that the more collective, you see, the more global is your conformational change, the easier is to obtain good overlaps with one of the low frequency motions. Otherwise, if you are dealing with some proteins with very, very localized conformational changes, the overlaps are not so bad, are not so good. Okay, so again, another conclusion from here is that elastic network models work better for global conformational changes, where you have, when you have localized transition, elastic network models maybe are not the right choice of modeling. So now we have talked a bit about the vibrational modes of proteins, which lie in the sub-THz range and which are informative of the biologically relevant conformational transitions of proteins. Now let's put dynamics aside for a second and let's see about perturbations. So in the early 21st century, there was some research group that tried to simulate the biologically relevant conformational transitions in proteins, so the conformational changes, by applying some perturbations at specific location in the structure. So this was, I think, the first study in which they applied a perturbation in a certain point, which is related to the binding phenomenon, and they tried to simulate the closing mechanism of this protein, ferric binding protein. So you see that the protein here is in the open conformation. It has also a closed conformation, and the conformational change is represented by this arrow, these green arrows here. But then what happens is that if you try to simulate the response of the protein upon a perturbation, which is this yellow force here, you get a response, which is this red vectors here, that is very well in agreement with the observed conformational change. Okay. And this approach was carried out by using molecular dynamics, cross-correlation metrics, and linear response theory. But this was the first time that they were trying to see the conformational change as something triggered by some perturbations in a specific part of the structure. In this case, they applied the perturbation in the residue, which is the closest to the, in this case, the iron atom, which is the atom that binds the protein when it changes the conformation. Of course, this is very easy to implement into an elastic network modeling framework. So other groups in the following years applied this static perturbations approach into the elastic network modeling technique. So what you do into this framework is very simple. It's very simple. It can be done with this simple equation where you simply equate the vector of external perturbations, so the vector of external forces, to the internal elastic response of the protein, which depends again on the Hessian matrix of the network and the resulting response of the protein, so the vector of the resulting displacement. So once you apply a perturbations to the network, you can very easily identify the response of your network, so the displacements of all your atoms, by using the so-called Hessian pseudo-inverse, which is again, as you can see here, computed based on the eigenvalues and eigenvectors of the matrix. So what this group tried to do was to simulate the observed experimental conformational transition of this protein, FBP, by applying a perturbation, again at the same residue, but by using the elastic network modeling technique. And you see that they were able to find some good correlations between the computational results and the experimental displacements in the conformational changes. And this time they were able to find the conformational change by applying the perturbations both in the holo conformation to trigger the apo conformation, or vice versa. So they took the apo conformation and applied the perturbation to the apo conformation to trigger the holo conformation. So this is basically to say that you can apply static perturbation to specific residue in the protein and can trigger the opening or closing of your protein. Then we just need to add another piece of information to end this introduction and to finally dive into the main topic of this talk, which is applying random perturbations all over the structure. So this is the results of one of our latest work, where we try to apply random forces, random perturbations, and again, we are talking about static perturbations here. There is nothing dynamic again going on. I was saying we were applying random perturbations, random both in directions and amplitude, all over the protein structure. So we take a protein structure, we build an elastic network model, a coarse grain elastic network model, we apply some random forces, so we are able to build a random force perturbation vector, and we simply evaluate the response of the protein, so the resulting displacement. Now at this point, what we did in this study was to compare the resulting displacement of the protein, not to the conformational change, because that wasn't our goal at that time, but to the B factors of the protein from the experimental B factor from the PDB file. So in order to have an idea if this perturbation-induced response agrees well with the experimental flexibility of the protein, which is kind of included into the experimental B factors. Well, we ran this analysis for a variety of protein structures, for 1,000 of protein structures. And yes, we found that overall application of forces all over the structure leads to, on average, a good prediction of the experimental B factor of the proteins. And also we found that if you apply the forces only on the external surface of the protein, the correlation with the B factors also increases a little bit. So basically we concluded from this work that application of forces to the protein structure is able to give you some insights into the protein flexibility. So this is it for the introduction. Sorry, it was a bit long, but I needed all these points now to go into the main part of the topic. So let's summarize what we have seen so far. So we have seen that both experimental methods, such as Raman and THz time domain spectroscopy, and computational techniques, normal mode analysis and elastic network models, show that the main vibrational modes of proteins lie in the sub-THz range. Then we have also seen that these sub-THz modes often point in the direction of the relevant conformational changes, which are crucial for protein function. And then we have also seen that if you apply static perturbations either to selected residues of the protein or to the entire protein structure, you can have some good prediction of protein conformational changes and flexibility. And now the idea was to put everything together. So on one side, we had the free vibration normal mode analysis, when you have no perturbations, but you have simply the equilibrium between the inertia dynamic forces and the elastic forces. On the other hand, we have seen that you have the response from static perturbation. So we don't have dynamics, but we have perturbation. So the idea was to apply dynamic perturbations to the protein structure. So we want to solve this complete equation here. And then to do this, we also added some damping to the system, not to have some purely resonant conditions, but to see what happens also with when you have when you add some damping to the system. So at this point, we needed to choose a proper dynamical perturbation, and in order to see what happens if you apply perturbations to proteins at specific frequency, okay, so to have just specific frequency components of perturbation, we of course decided to apply harmonic perturbations. So because with the harmonic perturbations, we have just one frequency component per perturbations, okay? So what we are doing here is to apply harmonic perturbations, so continuously to the protein structure, randomly. So again, we exploit that concept of application of random perturbations all over the protein structure, okay? So this is basically the framework of this analysis here. And of course, the frequency of the perturbation is expected to play a major role. So we want to understand what happens if we apply perturbations in the same frequency range of the main normal mode, so in the sub-THz frequency range, okay? And now we see what happens. So these slides are just a bit of mathematical calculations. We can switch from the complete equation in which the fundamental unknown is the response of the protein, so the displacements of the nodes of the atoms, basically. And now we use a typical methodology that we normally use in structural dynamics. So basically we decouple the spatial and the temporal part of the solution. So basically our atomic displacement are seen as a product between a matrix U, which is the matrix of the modal eigenvectors, so the normal modes of vibration, times a vector of the so-called modal coordinates. So we basically see the total response, total dynamical response of the protein as a summation all over the normal modes of vibration, each of these modulated by the time-dependent evolution, which is called a modal coordinate, okay? So now we can solve the problem in terms of modal coordinates instead of response of the protein. Again, a bit of calculation, so I don't want to go too much into this, but we exploit some properties of the mass matrix, of the Hessian matrix, and here we also do some kind of strong assumption about damping, because damping is something not really clear right now for proteins, but we assume that damping in this problem is classical, meaning that this matrix here, so the damping matrix times the matrix of eigenvectors is diagonal. This is very helpful for calculations because it basically decouples the equations of the normal modes. So we are able to have a set of equations, each of those for each of the normal modes. So we don't have the coupling between the normal modes anymore, which is not true, of course. The normal modes are coupled in some way because of damping, but since this is not completely understood in a quantitative way, at least, we're trying to understand what happens, at least if you assume that the modes are not coupled. Well, in this way, it's very easy to solve the mathematical problem because we have an analytical solution for the time evolution of each normal mode, so the time evolution of the modal coordinate, which is made of two terms, of two parts. One which is called the transitory phase, which after a while just vanishes because of the damping, and one which is called the steady state regime, which is, as you can see here, it oscillates with the same frequency of the external perturbation, but there is also some phase shift in the response, and there is some amplification or de-amplification depending on the value of this amplitude term here. So here you have all the definition of the different terms into the mathematical equation, but I don't want to bore you too much about this. If you want more details, you can read this paper here or the fundamental book of structural dynamics from Chopra. And so basically what we do is we can numerically solve these equations to find the dynamical response of proteins upon harmonic force perturbation. And let's see the results. So basically what we do is we take a protein, okay, which is supposed to have two conformations, so an open conformation and a closed conformation, so we have a conformational change. We start, for example, from the open conformation, this is the case of LAO binding protein, and we build the elastic network model. Okay, so in order to solve the equation, we first need some understanding of the main vibrational frequencies and normal modes of vibration of this protein. So we first run the traditional free vibration normal mode analysis. So we evaluate again the frequency spectrum based on this modeling technique. Here you see the spectrum of the normal modes in the frequency domain, and you see that the frequencies are, as we know, in the sub-THz range, and in this case we found a low frequency, the lowest frequency equal to around 50 gigahertz. So we are in the 0.05 THz range. Then what we can do before starting the actual simulation, we can also compare all these individual normal modes of vibration to the conformational change of this protein. Okay, so we know the conformational change, for example, the open to closed conformational change, and we compare this with the individual normal modes of vibration. And this is the results. So you see this is the overlap for each normal mode with the conformational change. The maximum that we can get, as you can see here is 0.76 and corresponds to specifically to the first mode of vibration. In vectorial representation, you can see here the first normal mode of vibration. You see that there is a clear closing of these two domains in the protein, and here you have also the comparison between the conformational change of the protein, so the experimental one, and the first low frequency normal mode from the elastic network model. You see that there is a correlation. The correlation is not 100%. Again, we found 76, but still the first low frequency mode is able to provide some idea about the conformational change of the protein. Okay, but now let's go into the results of our calculation. So now we don't want to investigate individually the low frequency mode, but we want to apply random forces all over the structure with a frequency, with an harmonic variation in time domain in the sub-THz range. So here you can see, I can start this video. Here you can see the resulting motion. So basically what we do here is, as I said, we build the elastic network model, we apply some random forces all over the structure. Here we used 50 gigahertz as the exciting frequency, which is very close to the one of the first low frequency mode. And here you can see the resulting motion. Okay, so the blue structure, which is evolving, is the open form of the protein, which is triggered by this sub-THz harmonic perturbation. And in the same graph, I also show you the red conformation, which is the target one. So this is the closed form of the protein. And you see that very interestingly, we are able to harmonically capture the closing mechanism. So you see that in a periodical way, you get to the target very closely. Okay, so you meet the target very closely. So we can maybe start again the simulation. Okay. Oh, actually, I can run it here. Okay, so you can see here you have some other quantitative information about this motion. You see that there is a first phase in which you have a transitory. Here I plot the root mean square deviation from the starting protein conformation. So you see that there is a transitory. After a while, the protein enters into the steady state regime, oscillating forth and back with the same frequency of the external excitation, and meets the and it reaches very closely the target conformation. Can't we quantify this closely reaching of the target conformation? Again, with the overlap. So here you see the time evolution of the overlap, this time calculated between the vector of the conformational change, so the experimental conformational change, and the response of our protein based on the harmonic force application. Okay. At each time step. You see that constantly you get very, very high overlap values. So here, basically, this is a quantitative representation of what we are seeing here from the movie. You see that every once in a while you get to the target very closely. And this very closely, in a quantitative way, is 94%. So this means that this application of random forces all over the structure at this frequency is able to trigger the conformational change with very good agreement. Okay, so we are basically reaching almost exactly the conformational transition. And here, for example, you can see the comparison in terms of conformational change, in terms, of displacement between the conformational change, so the experimental one, open to closed transition, and the snapshot, let's say, of the trajectory, so the snapshot of the dynamic displacement at a certain time point. Okay, this is actually, the motion is harmonic, so you can say this, you can see this graph every 20 picosecond, okay, and every period, and you can see that the correlation is much higher now. So this is the correlation between the conformational change and the response of the protein when you apply perturbations randomly, and this, you see that the correlation is higher than when you compare the conformational change with just one normal mode. Okay, so this is the main point of this analysis. So if you compare the conformational change just with the first mode, at maximum you are able to obtain 76% of correlation. But if you apply this framework, okay, and you allow the protein to vibrate, putting together all the relevant modes, you are able to get a much better correlation with the conformational change. And also this is periodical, okay? You see that you are triggering the closing of the protein very well in a periodical way. Okay, I can go very fast about this. We also wanted to see how much this motion is linear or non-linear, meaning that you know that the normal modes are linear, so they have a fixed direction in space and they just go back and forth along that straight line. But when you start to mix all the different modes together for generating a resulting complex motion, this complex motion can be non-linear. So when you put your attention into a particle, for example, an amino acid or an atom, the motion of the atom can be very complex, so it can be non-linear. So by evaluating basically the angles of these all these vectors over time, we were able to conclude that, as you can also see from this movie, the motion is highly non-linear. You see some very important rotational components here. So the atoms are not going just in a straight line as in the normal modes, but they're searching the space of the conformation basically in a curvilinear pathway. So this simulation, okay, this simulation here was just applying one specific random force pattern, one specific frequency of the external excitation, and also, I didn't say that before, one specific coefficient for the damping of the modes. Here we also had the assumption that all the modes have the same damping, which is not true, of course, but this was just to avoid a complex evaluation of the damping in this phase of the research. And you see that the value of damping here was set equal to 0.01. Okay, so 1% of damping for each mode. So now we also carried out a parametric analysis, seeing what happens when we change the frequency of the external perturbation in the range 0.5 terahertz. What happens when we change at least 3 scales of the damping, but still you see that we are in the underdamped regime, and this is also something we can talk about later. And also we wanted to see what happens if we change the direction of the specific force perturbation. So here in these graphs you can see all the different colored curves refer to 100 different simulations with 100 different force random patterns. Here I plot the so-called amplification, so the dynamical amplification of the displacements measured in terms of RMSD, of maximum RMSD, which basically provides an understanding of the potential resonance of the system. Okay, so you can clearly see some resonant peaks, and not weirdly, I mean, not surprisingly, these peaks are in the low frequency range. Okay, so you are able to induce quasi-resonant condition if you perturb the low frequency normal modes. When you on the other hand, you apply perturbations in the high frequency part of the spectrum, you don't have some amplification anymore. So this is one of the reasons why the low frequency normal modes of vibrational proteins are so important, because you just need a very little force to induce a very high displacement, as long as the force has a frequency which matches a bit the normal mode of vibration, the frequency of the normal mode of vibration. And then from these graphs here, you can also see the effect of damping. The higher the damping, so the bigger the damping coefficient, the lower the amplification. Okay, so but this is common. So when you increase the damping of your system, the displacements become lower. Okay, but I think this is the main figure of this webinar, because this is the parametric analysis, but in terms of maximum overlap with the conformational change. So here we want to measure how much we are able to reach the target closed conformation from 0 to 1 when changing the frequency of the external perturbation, when changing the damping of the system, or when changing the specific random force pattern. You see that immediately from this figure, when you trigger the protein in the high frequency part of the spectrum, the overlap with the conformational change is pretty low. Okay, so you are not able to actually triggering the right displacements. Conversely, when you apply perturbations in the low frequency part of the spectrum, you always get very high correlation. So this is that the famous 0.94 overlap. Okay, you can also take a look at this graph here, which is the same information but in a different representation. So here you can see the effect of the damping and the different force pattern in terms of maximum overlap. You see that almost independently on the specific direction of the perturbation, you are always able to get a very high overlap with the target conformation as long as the frequency of the perturbation is in the low frequency range. You see that the frequency is not always the same because it depends a bit on the specific direction of the forces, but you see that you are always in the 0.0 to 0.08 terahertz range, which is exactly this region here. So as long as your perturbation is here, which is the region of the main normal modes, it is likely that you are getting a very good correlation with the conformational change. Okay, so this was the main results of our simulation. In the few minutes that I have left in this presentation, I want also to show you what happens if we try to simulate the opposite conformational change, because so far we started from the open form of the protein and tried to, you see from this simulation, we try to see the closing. What happens if we do the opposite? So we start from the closed form of the elastic network model and try to trigger the opening. Well, as you can see here, the results are worse. They are worse even if we start by comparing the individual normal modes, and we have already seen that in the previous slides. So if you compare the single normal modes with the opening conformational change, the maximum overlap you get is, in this case, 0.56 for the third mode, which is this one. You see that the correlation is not very good. And even with our dynamical perturbation approach, the maximum overlap that you get is 0.75, something around here. And you see that, of course, it's higher, again, than when you compare the individual normal modes, but it's not extremely high. And this is related to what I told you before, that elastic network models are not a good choice if you want to simulate the opening mechanism, okay? But this is something that the authors at the beginning of the 21st century already realized. And also, in slide 19, so the previous slide, we also see the difference between a local and a global conformational change. So we were also interested in seeing if we are able with this method to trigger a localized conformational change. Well, unfortunately, again, the results are worse. So if we try to focus on another protein, this protein here, which has a very localized conformational change, you see here the blue curve, there is just a minor portion of the protein that moves during the conformational change. Well, if you compare the individual normal modes of vibration to this conformational mode, in this case, the maximum overlap you get is only 0.27 with the 14th mode. This is the 14th mode, and you see that the correlation is basically, there is no correlation, no meaningful correlation. And as expected, if you apply this framework of perturbing the protein dynamically, we were able to get a 0.44 overlap, which is not good. Of course, again, it's better than this one, but it's not good. And again, this is probably related not to the methodology, but to the modeling technique of the structure. So because we are using the elastic network model to try to get this localized conformational transition, which is not something very easy to do, as we have seen. So this is the conclusion of this presentation of mine. So we have seen that if you apply random harmonic perturbations on the protein elastic network model, you can be able to trigger some biologically relevant conformational change, but as long as the perturbation is in the low frequency sub-THz range. We have also seen that the resulting motion is highly nonlinear and captures much better the target conformation than when you consider only the individual linear modes. We have also seen that the specific direction of the random perturbation does not play a key role on the results. The key role is played by the frequency of the perturbations more than the direction of the force application. And we have also seen that probably due to the capability of the elastic network models, we are more able to capture the open to closed conformational changes, which are global, collective, rather than the opening mechanism, so the closed to open conformational change or the very localized conformational change. And then we have also seen that the perturbations at low frequency and with very low damping coefficients can also give rise to very high displacement with very high amplification, highlighting potential resonant phenomena. This is just a final slide concluding with a lot of open questions out of this work. So the first one is of theoretical nature. So, okay, we have seen that if you apply harmonic random perturbations at some frequency, sometimes you are able to trigger very well the conformational change. Now the question is, what do these harmonic perturbations actually represent? Well, one explanation can be that forces onto the protein structure can be a representation of the interaction between the proteins and the external environment. So for example, collisions with solvent. And investigating only the harmonic perturbations, we can try to see these harmonic perturbations as the frequency-dependent components of these time-dependent collisions with solvent, but this is still under exploration. Or this can also be related to some low-frequency sub-THz electromagnetic or acoustic perturbations. But as I said, this is still a very important open question. And there are other open questions that we're trying to address in future future applications of this work. In the sense that we are trying to see what happens if we model the structure not with the elastic network model, but with other techniques. In this case, we are hoping to get also good results in the bad cases, so for localized conformational changes or for the opening mechanism. Also, we want to understand what happens if the dynamic perturbation is not harmonic, but is just, for example, a pulse, very short in time. Or what happens if the perturbation is not applied all over the protein structure, but just, for example, on the surface, or just to few selected residues or representative regions of the protein. And also the last, but not least, open question is damping. So how much damping actually influences the protein response, which is related ultimately to the question on the nature of low frequency modes. Are these modes overdamped or underdamped? As you have seen in our work, we have carried out our calculation in the underdamped regimes, but this is not completely the case. So we are not so sure that all the normal modes of vibration are underdamped. So there is still a lot of research to do about this problem here. Okay, I think I'm done here. You can also take a look at all the references that I put into this presentation for the figures and the other equations or so on. And specifically, this work was taken by this very recent paper of us, which we published last year. Okay, thank you everybody for your for listening to me. I hope you enjoyed the presentation, and I'm open to your questions.
Thank you very much, Domenico. It was really, really interesting. Thank you. So I have a list of questions, but I usually try to give a opportunity first to the attendees. So if there is any question from attendees, please raise your hand and I can unmute you. Oh, I see. Patrick Sanet is here. Nice. So Patrick, I will make you a presenter so you can unmute yourself. So go ahead. We can see you, but cannot hear you. You also have to unmute.
Can you hear me?
Now we can. Yes. Please go ahead.
Okay, thank you. Sorry. Thank you, Domenico, for this nice seminar. I have a lot of questions, so I will limit it myself to some of you. First, in your simulation, the Hessian is constant, right?
Yes. Yes.
Okay.
So it's just one evaluated on the equilibrium structure, so from the crystal structure. Yeah.
So you could use the green function, the response function, which are poles to the normal mode frequency to simulate the displacement, right? With some damping. Okay.
Yeah.
Now I have a remark about the close to open conformation. Is it also possible that it's not a ENM, which is a bad model, but the fact that when it's close, the system maybe needs more modes to do this, to the conformation from the close to the open.
That's
because in the open, the B factor in general are larger, so one mode is dominant. Because the mode with the largest eigenvalue is also the solution of the variational problem, and maybe in the case of the close to open, you need more modes, and so you should work maybe in a multidimensional space to see the overlap changing as a function of time. No?
Yeah, in a way, this is already this should already be captured by our model, because maybe I can go through the math. So when we apply forces in over time, we actually look for a response which is already a summation of the mode, and so we are basically including all the modes into the calculations, and each of the modes is weighted by a specific modal coordinate. which arises from this equation here. So we are actually considering, all the modes into the response. In my opinion, what happens here is that when you have a closed conformation with elastic network models, you generate much more connections because the conformation is more closed. So with the same cutoff, for example, you have a lot more connections. So it's much easier and much more difficult to kind of see the opening of the mechanism. Another thing that I wanted to say here is that this work was done in a very brute way, let's say. So we didn't optimize any parameter. So we use the same cutoff for the generation of the elastic network model. This is basically an isotropic network model with all the spring constant, which are equal. So one thing that can be done is to optimize a little bit the parameter to try to give more freedom to the closed conformation to open. Because if the model is already constraining too much the protein, no matter which kind of perturbation you give to the protein will never open up. So yeah, I agree with you that you need more modes, but I'm pretty sure the model is playing a very big role into the not capturing the opening.
Yeah, I agree. But what I mean also is that maybe the vector, the experimental vector. Sorry.
Okay, sorry. Okay.
Yes, yeah, there was a cut. I don't know why. So I mean, the experimental vector from open to close and to close to open, maybe they're not the same. Maybe if the phenomena from close to open as to cross a barrier or as to, you see, we observed that in one case for HSP70, which is a complicated protein, but for sure the transition from open to close and close to open were not the same. Of course, we have only the two experimental structures, so we can build the same vector, but maybe the transition is not the same.
Yeah, you're right. The pathway is supposed to be different. So this is another point to.
Okay, thank you.
Thank you very much, Professor.
Thank you.
Okay. So yeah, you were quite economic in the questions, Patrick. So that's great.
Yes, I try.
But maybe if there's more space, let's try to have more discussion. So maybe I'll take opportunity. So you answered most of the well, maybe the most general question, which I was curious, Domenico. Why did you choose these kind of perturbations, like a harmonic in time, but randomized, I guess, both in direction and amplitude, or what was randomized? So what does this correspond to physically? I mean, why did you choose this kind of perturbations?
Okay, I can actually answer to you by, so we actually started from this. So the idea came after this work. So at first we were trying to just simulate the protein flexibility. Okay, this work was published later, but it actually was done before.
I see.
We started from this idea of applying forces all over the structure randomly, but in a static way. So there was no dynamic.
Okay. It's related to B factor and flexibility.
Exactly. Try to simulate it. Then we try to see what happens when we put this framework into dynamics. So apply the perturbation dynamically, and and that was basically the reason why we try to apply forces randomly in both space and in direction and amplitude. Why I applied forces harmonically in time? In order to separate the frequency component. So because if you have just pure harmonics, you can just take a look at the frequency. If you apply whatever other form of signal you want, you will have more frequency in your more frequency values in your signal. So it's more difficult to understand if the response is triggered better. Is it just because that specific signal, or does it have to do with the frequency or with the specific part of the protein which you perturb? So the idea about application of harmonics was just to separate the frequencies, 1 THz, 0.05, and so on. And now the idea, of course, will be to put more frequencies together, maybe. So to try to simulate what happens when you apply pulses, for example. So like short pulse, which have a strong component in a frequency, but also have some, for example, higher harmonics. So yeah, that was just it, basically.
Okay, I see. So it came from more from flexibility modeling than from, let's say,
yes,
some particular perturbations which could be applied by external forces, because those would be probably more vectorial. Like you can imagine, if you have an electric field vector, it's something we are considering in our situation.
Mhm.
And basically, depending on a charge of a particular grains or atoms in the model, they will be either pushed one way or another, but all, let's say, in a collinear way. So it'll be not anymore random in orientation, but the strength of the perturbations will be given by the charge of the grain, and the direction will be either parallel or anti-parallel to the vector. So it will be still randomized in ways, because there is kind of a random distribution of nature of charges. So it's kind of corresponds, maybe more than I thought, to what we are
actually the original idea before this application of static forces all over the structure was to, I mean, the idea was just to include something that can be representative of solvent interaction into the elastic network models. That was the main idea that we had when I was in Jernigan's lab. So to try and simulate the solvent some way with the elastic network models, because elastic network models are really cool. I mean, I work with them daily, but there is just mass and stiffness. So you cannot play a lot more other than these parameters. So we were trying to see if the effect of solvent can be modeled not by putting more into the model, like in terms of modeling, but by just an effect onto the model. So like a perturbation. So
I see. So I see basically modeling interaction with something external. In this case, it will be the solvent. Of course, then probably, yeah, I see. So yeah, I understand. This is kind of the first steps on a journey to model that.
Yeah. And also in that analysis that we carried out with the B factors, we tried to see what happens if the perturbations are still randomly distributed in space, but only on the external surface of the protein. Because if you think about that, if that force is related to a solvent effect, doesn't make sense to apply a force in the core of the protein. So it should be, I mean, the effect should be maximum at the surface. And so that was the logical flow
I see.
During this journey, but yeah.
I see. Okay, very cool. I also have some few technical questions. So regarding the damping, of course, this is a big question in spectroscopy. People are even fighting about what's the reality. Did you try just one, because it should be easy in competition? What would be the behavior of the models if you start to approach? Oh, we're getting some storm here. I apologize for the noise. If you try to reach slowly the the threshold of the underdamping, so it would be probably damping coefficient 1 or quality factor 0.5, however they have defined.
No, we didn't try that. We still remained in the underdamped limit, just because that wasn't the focus of the work at that time. But still, this is of course something I was planning a while ago, because it's easy computationally. So you can just go right there. One of the problems that I think there can be, not computationally but theoretically, is that another limitation of this work is that we are applying the same damping coefficient 0.01, 0.001 to all the modes. So if you say, okay, let's push it to the critical damping, do we have to push it all the modes to the critical damping or just the low frequency? So you see, it's very tricky, because imagining that all the modes have the same damping was the only choice I have at the beginning, but it's absolutely not right. So I don't know
I agree.
It makes sense to push all the modes toward the critical damping, because I really expect that the high frequency one are underdamped. So they want to jiggling around a lot, and maybe the low frequency one are overdamped maybe, or critically damped. So maybe a more distance between the more distant distribution. So it's very difficult. But yeah, this is definitely something I will try at some point.
Yeah, that's really interesting. Yeah, that's a very crucial question, I think, for any potential resonance responses to take place. Yeah, great. So I maybe something which Patrick Sennett asked, but in those analyses where you try to see the let's say the overlap with the biological motions or conformational changes with the particular modes, I mean, you kind of addressed that. So if you could instead of having just comparison to single modes, for example, you had the 14th mode for one of the proteins.
Yeah, with one protein with localized transition. Yeah.
So yeah, maybe I think Patrick mentioned it too, that instead of trying to see, because people claim it based, it depends on what method you use, that if you rather use the sum, sum of the vectors, like not only first one, but the corresponding to, let's say, sum of the motions of the first few modes. So that's something which could be giving better results. So I'm not sure if you did the summation or.
Yeah, no, you can do the summation. Basically, you have to calculate the integral of all these little bars here, and you take all the contributions of the mode, that at some point you will reach some, I don't know, 0.8, 0.9 overlap. The problem is that you see a distribution between this and this is very different. So this is a very collective motion for this protein, and this is a very local one. And this already tells you that, I mean, if you want to stick to the low frequency modes, for localized transition is very, very difficult for elastic network model to capture. But this was already realized 20 years ago. So this is not actually something I'm saying today. This is the seminal paper from TAM. They already realized that it's very difficult. So I think, again, in this case, it's mostly the modeling technique, probably. But
I think it's also protein dependent. Like there are some proteins which are behaving more collectively in their functional modes, and some which have to sum up the many. But Patrick, you had some questions. Go ahead.
Yeah. Yes, I have another question. It's not related directly to this, but I agree that some proteins will be single mode and some proteins will be multiple mode. And we observe that in protein folding also. In protein folding, we could use PCA to show that one mode was driving the folding and other units 5, 6 mode. But my question was another question. Did you try to introduce a thermostat? Because you could add Langevin force to your equation, and then you could simulate the thermostat and see all the signal change with temperature. That would be, I think, interesting also.
Yeah, no, this I didn't do. So this is another thing. Of course, I can think about it, and probably would improve the results for these bad cases. Let's see.
I don't know. You are still in the harmonic regime because your SN is harmonic. But, but maybe it's also another source of noise, which can be interesting. Okay, thank you.
Probably it's also because I mean, it's not completely true to say that. I mean, we know that normal mode analysis works in the harmonic regime, so usually it should be applied only for small scale vibrations. But still, it was realized. I read some recent papers about this, that if the motion is collective, you can still use the elastic network models and the normal modes to go to that extent. As long as your mode, as soon as your conformational transition becomes localized, even if it's very small, the harmonic assumption basically is losing its power. So at that point, you need to, I need to include some anharmonic effects into the Esham matrix, because I'm not expecting to simulate that conformational transition just with the Esham matrix evaluated into the equilibrium crystal structure. So definitely, yeah.
But I would expect the reverse, because the low frequency mode have a large amplitude. So I expect to be more anharmonic than the high frequency modes, which are much more restrained. And so
yeah, but I am not sure about that, because I read some papers where they try to estimate the anharmonicity of the transitions with the low frequency modes, and it seems that the key player is the collectivity. So even if your mode is high amplitude, high displacement, as long as it is collective, the harmonic approximation is good. If your mode is very local, even if it's small, you have anharmonic effects that play a big role. But
because I remember there is a paper by Go a long time ago. It was not normal mode analysis, but was PCA, principal component analysis, and the low frequency mode were multiple Gaussian, multi-peaks. So they were not
PCA
harmonic
on the crystal structures, on the MD trajectories.
MD simulation, and so they were not harmonic. And the high frequency mode were single Gaussian.
Irrespective of the collectivity of the motion, so as long as the motion is low frequency, they are harmonic.
Well,
so
at least at least I think in this paper they didn't compute collectivity.
Okay.
Because it's an old paper.
Okay.
But yeah, but this harmonicity is an interesting question to investigate. I don't know. I don't know the answer.
I'm not completely sure about that, so I need to look more into details about this. But yeah.
Thank you.
Thank you.
Okay, any more questions for the audience? Yeah, I mean, probably the questions I would have would be following up of related to the what if. I'm not sure if you have those data then. So, well, I guess that this frequency selectivity would be if you approach critical damped situation. Let's assume having the same damping for all the modes, then I guess this frequency selectivity would slowly die out, right? I guess. I'm not sure.
So you mean if I approach the critical damping for all the modes, I don't see any differences in the response when I change the frequency?
Naturally, I would guess that, but I'm not sure now. It depends
Probably.
really on a.
I really don't have an answer for this. I should try, but I think I can try. I can try very quickly and, and tell you. It's just a matter of putting C
Changing
equal
the
to
number,
0.99
yes.
for every mode, and let's see the simulation.
Yeah, I see. Okay.
I can put a quick response in the YouTube video, maybe.
Yeah, well, that'd be cool. Of course. I have one more general question. That's probably something which protein people are really asking as well. I haven't seen many papers, maybe only one on that, but I would like to hear your opinion regarding the, what is the role? These general claims, which we are all so interested in, that these fundamental modes, at least in certain proteins, seems to be highly correlated with the biological motions. But this is something which people kind of start to, let's say, believe or agree on. But what about the time scales? The effective time scales of these motions are usually milliseconds, even longer, or microseconds, depends again on the protein. But the time scales from a harmonic, let's say, approximations are, well, picoseconds or nanoseconds, maybe to a fraction of nanosecond. So how do you perceive like the connection of these? I mean, one of the maybe one most typical thing how I understand it, that these motions are kind of helping to cross over the barrier between the, let's say, closed and open states, or whatever are the two states of the protein. So is this the perception also you have, or what is the like connection of these things? On one hand, very high, let's say,
frequency,
very high frequency, let's say very short time scale motion of the same shape, so to say, the same motion, but the biological time scales usually are much longer. So how do you perceive this connection of the eigenmodes compared to the biological motions?
This is actually a question that I ask myself every day, because even from my calculations, all the papers and all the things that I've discussed, all the normal modes, from one side they are connected to the biological movements, and from the other side they are in the picosecond range. You see, because you have Raman spectroscopy, you have THz time spectroscopy, you have normal mode analysis. A lot of computational and experimental stuff says that these normal modes are in the picosecond scale. And we know that these big domain motions happen in the millisecond or even seconds time scale. So yeah, one idea can be related to damping, maybe. At this point, maybe it's the damping that is kind of preventing this very fast oscillation, but is maybe allowing just one of these oscillations to pass over this damping filter over, I don't know, 1,000 of tries, for example. Or as you said, it's just a potential motion that the protein has in itself to overcome the barrier, and just once in a while it is able to jump, and it takes maybe 1 millisecond to do that. So it starts oscillating in the picosecond time scale, and when there is something that is able to add the extra energy to jump the barrier, maybe he can do it. But I don't, of course, have the answer to that. I mean, it's also something that really annoys me sometimes, because I cannot see an explanation for this myself. So
I think it's really an open question for it. It's not like and people are basically asking this. I think I recently found one paper was trying to connect these things. Maybe I can send it to just to for thinking. But yeah, it's something very interesting. Okay, great. So do you have any more questions?
Another reason can also be that when we look at proteins, we only try kind of focus on the protein and neglect everything else, you see? And maybe that everything else is very important. So even neglecting all whatever, I mean, all the other stuff that are playing with the protein that we are focusing on maybe are also important for, so I don't know. There is a lot.
Brings me to, yeah, that's interesting. This brings me to a question, actually, what we are just right now, we are analyzing in our full atom simulations. Well, most of the spectroscopists will tell you that, or even the people who are biophysicists for proteins, that water matters a lot, right? Obviously, not only as a passive solvent, but basically kind of interplay of the motion or fluctuation motions of the water molecules and proteins kind of determines the function of the protein. So along this line of thought, did you consider, or did you maybe try to include to the NMA also the water molecules around, like few layers?
No, in this simulation I didn't. We did in our static perturbation simulation. Okay. So when we apply forces on the elastic network model, we also included the layer of the water molecule, the explicit water molecule in the PDV. But in that case, we didn't have any time information. So we just wanted to see how the fluctuation of the protein, how the response changes when you also add this extra layer of connections with between the protein structure and the water molecules.
I see.
So yeah, we did on a level, but not on I mean, in this, not from a dynamical perspective. So yeah,
I see.
This can be another interesting thing to do. See.
Absolutely. So, and did you in this case, did you try to compare how the response to the static perturbation would differ if you have the layer of the water or not?
Yeah, we found. I mean, it is in that paper, and we found that when you add the extra layer of water, what?
Sorry for asking you something you published. Just thanks.
No problem. No problem. Absolutely. So we found that if you add the extra layer of water molecule, you are able to capture better the flexibility. So it's already something real. So the protein layer, the water layer, also has an effect on the intrinsic protein, the intrinsic way of the protein to move. But yeah, this again was just from a static perturbation perspective, not on the dynamical motion of the protein.
I see. Yeah, that's very interesting. Okay, maybe one more thing which also we are trying to probe. The starting point, the structure matters a lot for, yeah. So did you try to play around, like maybe having a sampling, but basically what we are trying to see now for, let's say, for inputs to our NMA, not to have only the single structure, but actually run the MD long and have like many exactly multi-ensemble, but basically ensemble of the structures or yeah. So did you try something like that too?
No, I didn't.
Okay.
I just, as I told you, this was just a very brute work. So I just took the structure, build the elastic network model, and just shake it like a building. So and see what happens.
Okay. Yeah, usually for buildings there is one structure. Usually,
yeah,
I mean, if it's one building, it's one building. For proteins, the starting structures are so diverse. Yeah, because it brings me to what Andrea Markels was showing in our webinar, and they have also some nice papers on that. That they had to capture, or let's say to recapitulate this experimental terahertz spectra. They need to make this ensemble analysis, because basically what they found is that at every certain point of time there is instantaneous terahertz spectrum. And this is what you get in reality is kind of ensemble. It's an average of those things.
But this is for from the computation, you are saying?
For computation plus when they were comparing to experiment, their experimental data. So they had to do computationally, they had to do kind of ensemble averaging. But I see Patrick has some maybe comment on this or something similar. Go ahead, Patrick.
Yes. In fact, we have some experiment in the lab, which are some published, some not published, not by me, but by one of my colleague. They are doing single Raman spectroscopy of proteins, and in fact, it's exactly what you observe. Every spectrum is different because the protein is fluctuating from one local minima to another, and you have different spectra. Of course, what they observe, they are not observing the low frequency modes, which are probably not so different, but all the other modes, which are high frequency, like amide band, I think it's about 1,600 centimeters -1 or something like that. These modes are very sensitive to the conformation. I don't know about the low frequency mode because there, to my opinion, there is no experiment. We did some calculation, which are, I think, not published, where we observe also some fluctuation of the lowest frequency mode. I mean, the frequency and the eigenvector are slightly different from one conformation to another, and the conformation were just catch from MD simulation, and we minimize the energy and we compute the spectrum each time. So we see some fluctuations, but maybe the most important fluctuation are maybe the high frequency, I don't know. But for sure, experimentally, I confirm that we have also experiment here showing that.
But sorry, when you say change in the spectra, are you referring mainly at the shift in the frequency? So you see the peak, which is shifted along the frequency axis, or just the amplitude?
Both.
Okay, so
experimentally both. Yeah.
Okay. Okay. So
and in the calculation also. Yeah.
Okay. I see. Okay. Thank you.
Okay. Yeah, it'll be actually very interesting because if we would compare the sampling from MD, let's say to have many starting structures, then it'll be actually very cool to have some coarse grain approach to calculate NMA, because if you are doing it for full atom, maybe we could skip that and maybe NMA could go to, well, sorry, the elastance model maybe could go to a good approximation if it takes into account all the reasonable phase space of the conformation. So that could be a way to try. Yeah. Okay. Yeah, that seems to be a very interesting discussion, but I think we should proceed. But definitely, I think our webinar will be also home to other protein dynamics talks. So I think if there are no more questions, I would like to thanks to our today's speaker, Domenico Scaramozzino. So it was very interesting, and definitely we'll keep in touch for a potential future collaboration as well. And I also thank very much to our attendees, particularly now today to Patrick Sennett for having very good and insightful questions. So I think we also should keep in touch, maybe all three of us to try to develop some interesting things together. Okay, so thank you again to everyone. And now I'm going to stop recording right now.